Now let's discuss about translation and gene expression using a YouTube video too. So transcription and translation. I'm going to show you a movie which is about transcription, which is a synthesis of mRNA molecule from DNA template. And that transcription begins with when transcription factor interact within regions of DNA, there's a promoter. And the promoter is a specific DNA sequence that facilitates transcription of a specific gene. So let's talk about this movie.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. The innermost workings.
Now, while you watch this nice, interesting YouTube movie, and that is very interestingly made, however, you shouldn't consider it's actually just true. There's some of the imagination together, but it's very interesting to watch how things happen. So in the end, the gene expression is what we want to understand. So all the cells in our body contains the same DNA information. However, each of the body cells, such as skin, such as liver, such as lungs, they all look different and they function different. So where comes the difference? It actually is coming from a gene expression, because they do have the same blueprint, but the expression of the genes are different. That makes all these diverse kinds of cell types or tissues and based on this gene expression. So while all the human cells contain the same genetic information, why does the cell of each tissue are different with their unique properties? And that we call gene expression, and using genetic information to synthesize functional products, often proteins. And how to control this gene expression is the key for all different kinds of cells and their functions. So let's take a look. So from the chromosomal DNA, and transcription makes primary RNA transcript, and by joining exons together and eliminating introns, this is called RNA processing, create those messenger RNAs. And that messenger RNA came out of the nucleus as a RNA transport, and this mRNA can degrade or it can be translated. This translation allows them to make a growing polypeptide chain, which is a building block of a protein. And that protein will be also folded and post-processed, and that generates a folded protein. And many of these proteins are unactivated, and if once it can activate it, they can perform more a function, such as an enzyme. So how to control these gene expression in individual cells is very important to control. And once we know, we can even control them too. So that level could be DNA, RNA, and protein level. Let's first see from the DNA level control. So we know that there's a promoter regions of genes, and that can be bind specific transcription factors, which can either enhance transcription or inhibit transcription of a gene at the DNA level. Once we create an RNA, at that part also there's a control, which is endogenous control, and even we scientists created exogenous ways of controlling this RNA for controlling gene expression. So first, endogenous one called a microRNA, which is a very short sequence of RNA, and that has a controlling function, or we call it as a target degradation of specific mRNA transcript. It actually, microRNA is a single strand that bind and that prevents it from translation. Or another aspect is called so-called siRNA, or small interfering RNA, which is exogenously made of human make this, and you can use this siRNA. You can put it into the cell, and this can interfere a specific gene to become translated. So this is revolutionize our genetics, because now by using this siRNA, you can have a target gene, and you can specifically silence this gene to be expressed. So this is a great tool for genetics aspect to find out what's the function of a gene if you silence them and unsilenced one, you can compare the role of this. Also, gene expression can be controlled at the protein level because protein can be inactivated or compartmentalized until they are needed. So next time we will discuss about DNA mutation. Thank you.